Welcome student. In today's class, we are going to study the fungi. Unit number 4. In that, we are going to study the first point, that is the mucor, or the first genus, that is the mucor. It comes under the kingdom fungi. And then we'll see the taxonomic position of the mucor, or we can say the classification of mucor. So, Mucor comes under the kingdom fungi, division Mycota, class Zygomycota, order Mucorales, family Mucoraceae, genus is Mucor. The next habitat of this Mucor, it appears readily in damp horse dung when kept under a bell jar. It is present in small soil, bread, and other food materials. Mostly, these mucus are present on the bakery products, which are dead and decayed one. Whichever the food material, the bakery products, we kept for long time, and they get spoiled, and it occurs. The mucus will appears on this the bakery products. Okay, then the vegetative structure of the mucor. Here you can see the vegetative structure of mucor. So this is what the vegetative structure of mucor. So here the mycelium consists of numerous cylinder, freely branched filaments. These are called as hyphae. So here you can see this is the numerous structures, branched filaments. These are known as what? The hyphae form a fluffy mass, white in color, called as mycelium. So here the white color fluffy mass, this is known as what? The mycelium. Means the large number of hyphae consist of mycelium. After mycelium formation, reproductive phase starts. Then black pin like structures appears on the mycelium. These are called as sporangia. So here black structures, you can see these black structures, and here also you can see this black structure. This structure is known as the sporangia, and this sporangia produces the sporangia produces the spores, and this hyphae, these are the cenocytic and aseptate. These hyphae, these are what? The cenocytic. Cenocytic means what? Without septa or without cell wall, and the same meaning of aseptate. Then at the vegetative state, the hyphae consist of a stolon, horizontal hyphae, and rhizoidal hyphae. So somehow this type of hyphae means root like structure. This is known as the what? The rhizoidal hyphae. The rhizoidal hyphae helps in the absorption of nutrients from the substrate. So this is the rhizoidal hyphae. This helps to absorb the nutrients from the substrate. Means whatever the soil and dead and decayed material is present. So from that, these rhizoids, they absorb the food material or nutrients, and then the cell wall is made up from the chitin. So this cell wall of this mucor is made up from the chitin, and this cellulose is absent in this mucor. And the organelle like mitochondria, endoplasmic reticulum, etc. are present in the cell. So the organelles are present. The next, so you can see here the structure of mycelium. So this the single structure, we can say this is the hyphae, or the this is hyphae, and the large number of hyphae, this is known as what? The mycelium. So here you can see this is what? The cenocytic hyphae. So without cell wall, without septa, and here nuclei is present, vacuole is present. Vacuole means the open space. Oil droplets are present, cytoplasm is present, hyphal wall, and the cell wall is made up from which substance? The chitin. Here even here you can see the cell wall, vacuole, ribosomes, endoplasmic reticulum, mitochondria, plasma membrane, nucleus. So this is what? The structure of hyphae of mucor. Okay, then next, here you can see the structure, overall structure of mucor. So here the substrate mycelium, here the sporangiophore, the elongated structure, that is what? The sporangiophore, and the and the attachment between the these sporangia, this is known as the what, the septum, and the solid part, that is known as the apophysis. Here the columella is present, and here in sporangia, sporangiospores, spores are present. And here this structure is what, the wall of sporangia. This wall of sporangia is break. Here you can see here, it can break or rupture when the sporangium get mature. That time it get break or rupture and spreads the spores. So this is about what, the structure of mucor. Then next one is the vegetative reproduction. So reproduction in the mucor. So first we'll see the vegetative reproduction. So first, vegetative reproduction is takes place by fragmentation. Here you can see, when the separation of hyphae occurs from the aerial portion, then these fragments are capable of producing new hyphae, and this is called as a fragmentation. When you can see here, this hyphae, when they get separated from the aerial portion, or any absorptive portion, they get break and produces fragments, small fragments, and they these fragments are capable to produce a new hyphae, and this is known as the fragmentation. Then next one is chlamydospore formation. Under a favorable condition, the fungus produces special modified thick wall resting cells called as a chlamydospores. They contain food and they are very resistant to desiccation. When conditions become favorable, it germinates to form a new mycelium. So here the special thick cell resting cells, we can say that. So these resting cells are known as the chlamydospores, and when they get the favorable condition, that time they germinate to produce a new mycelium or new mucor hyphae, and produces the mucor, complete mucor. Okay, then next is the asexual reproduction. Asexual reproduction is takes place by sporangia and sporangiophores. So here you can see the structure of mucor. Here you can see the structure of mucor. Even here you can see the structure of mucor. It takes place by the formation of non-motile multinucleate sporangiophore. The sporangia arise singly and terminally at the tip of sporangiophore. The young sporangium is white, spherical in structure. At maturity, it turns into black color due to formation of spores. In the center of spore, there is a large dome-shaped structure is called as columella. The sporangia are haploid. They are produced in haploid mycelium and produce haploid spore. So here you can see the overall structure. Here these sporangia are what, the multinucleate and non-motile cells, and these are produced on the terminal at the tip of this sporangiophores. These are what, the sporangiophore. This is what, the sporangiospore, and this is what, the sporangium, and this sporangium produces what, the sporangiospores, this one. And here the columella, it is what, the dome shaped structure. Okay, then this is what, the aerial hyphae, that is sporangiophore. Here the prostrate hyphae, and this one is the inter one hyphae or subterranean hyphae. Then next, the spores. The spores are dispersed after the rupturing of the sporangia. They are dispersed by wind. So here you can see in this sporangia, the sporangiospores get sporangia gets ruptured, and spores get dispersed, and the spores are motile. The spores are motile, oval, and contain protoplast surrounded by single layered cell wall. And under a suitable condition, the spore germinates to form a germ tube. The hyphae arises from the germination tube and spread in all direction. So this is about the spore. The next, sexual reproduction. Sexual, it takes place by sexual reproduction is takes place by gamete and gel copulation. The fusion of gametes are multinucleated and are distinct from each other. So here you can see the positive hypha and negative hypha, or we can say the male and female hypha. These are produced, means they get fused, fusion of two gametes, and these are multinucleated, and they are different from each other. The sexual process starts with the formation of two special structure. These are called as a zygospore. So when they get here the male and female hypha fused, and then here when they fused, here they produce the progametangia. Then after progametangia, the gametangia is produced. When this gametangia is fused to form a zygospore, and this zygospore is what the formation of sexual, means the process of sexual process. Okay. Then they arise from near the apices of actively growing somatic hyphae of positive and negative strains, and they are chemically induced. The zygospore meet and adhere in pairs at the tip to form a fusion septum. Then the tip swells due to flow of nuclei and cytoplasm in them, and these are called as progametangia. Progametangia combine to form a prozygosporangium containing a many diploid nuclei. These are called as a zygospore, and this is when after this zygospore is produced to form a new mycelium or new mucor mycelium, and produces a new mucor colony. And here you can see the sexual cycle. Here you can see the vegetative mycelium. Here the vegetative mycelium is there. Here this positive and negative strands mix to form a progametangia. Then here the suspensor is formed. Then young isogametangia is produced. Then here the conjugation is takes place. Then here the zygospore is produced. Then zygospore is get germinated and produces what the microspores, and this microspores undergoes the meiospore germination, and it germinates to germinate and produces the new vegetative mycelium or new mycelium. So this is what the life cycle of mucor or sexual cycle of mucor. And this is about the mucor, life cycle of mucor, and mucor classification. So up to this you have to study. If you have any doubts, you can ask me. Thank you.